thank you for that lovely introduction. It's great joy to be here, and I'm immensely proud of GETA, and I'm saying that to the rest of the world because these are parents that change the world, and we've had numerous discussions about this, that you are the ones that can get politicians to get us DEE nurses around the country. You are the ones that can make the world change, and we're there just to help you. Congratulations. I think this is the biggest GETA to my eye, and there are quite a lot of people online as well, so I'm very excited about that. Now, Leah, who set up this program with the team, where are you, Leah? I've lost you over there. Very kindly, said I could talk about my sabbatical, and my secretary rewrote the title to say this because I travelled rather a lot. I want to just have one talk about the disclosures slide. I did that yesterday at the SYNGAP, the small SYNGAP meeting we had. And I think it's really important people look at these and go, oh, she does all this work with all this pharma. That's why they're bringing drug trials to us. And we have more of our drug trials team here today than we've usually had. And I'm excited about that because they're getting into the DEE space, the boutique trial space that Piero will talk about later, N of ones, and this is where we're really going to get treatments for these very rare genetic DEEs. This disclosure slide looks like, how do you do it? I don't really do it, they do it, but I really try to bring trials to us because it makes such a difference and it puts Australia on the map, and I think that's really important. Now, this is my sabbatical. Most people go for sabbatical for 6 months to one place or 3 months to two, but just to emphasise, this was not a holiday, and I'll tell you a bit more as we go along. It was a dream of mine to have a sabbatical, and I'd never had one, and I could only do it because of my colleagues. Many of you know that Sam Berkovic has been my partner in genetic epilepsy crime since my PhD about 100 years ago, and his role has now been taken by the wonderful Piero Peruca, an adult epileptologist who's interested in genetics. His PhD was in genetics, and we've got to get him into the DEE space, so it's great that he's sitting just over there. Many of you will know my wonderful EA, Natasha, who helps the world to go round, and I'm immensely proud of Briley, who's here, who was the first DEE nurse in the world. You'll be pleased to know that other people are copying this model already. There's one in Germany, and lots of people say, we've got epilepsy nurses. They're not the same as a DEE nurse who is someone who specialises in this area. Now, I could only have a break not from work, just by the way, because Dr Michelle Dang and Dr Annie Chu, who's here, managed my Austin work, and Dr Emma MacDonald-Laws, who lots of you will know from RCH, she managed my rooms, and I'm immensely grateful to all of these people for letting me have that space. But it's bigger. This is a lot more people to thank. First time I had missed our annual research retreat in 30 years. I wanted to thank the team to allow me to do this, and you'll see lots and lots of different team members named here. So what did I do while I was not on a holiday? I completed this big review of DEEs with colleagues. Samir is the mentor of Andreas. Heather is in the US as a geneticist. Renzo, I'll come to later, is an Italian. And Amy is a wonderful young paediatric epileptologist in London. And so I'm going to define DEEs. Some of you who've heard me before will go, oh gosh, this again, but we just saw there are a whole lot of new people in the room. It is a complex concept, and even though we've written about it many times, people still don't get the essence of it, so I do spend time on it. And it basically brings together three concepts. Etiology, which is, I see, so my thing's not where yeah, it is. Etiology, which is the medical term for cause, epileptic activity, and developmental impact, and that could be developmental slowing or regression. If we just want to think about this term, break it up into two bits, into epileptic encephalopathy, where we just heard from Mel with her beautiful story that she told us about the gorgeous Stella, that she presented with this seizure type, which are epileptic spasms. And this is one of the prototypes of an epileptic encephalopathy. You have a baby that develops these little spasms, very brief, they don't look too bad to start with, but it's typically associated with regression or slowing, but more commonly regression. Has a very typical EEG. And I just wanted to stop here for a moment to talk about this little girl. She's an identical twin, and we saw her with her seizures. We did an EEG also on her identical twin, and she was developing epileptic spasms. She had the abnormal EEG, but hadn't yet had seizures. So clearly genetic, identical twins, identical epilepsy. We sent her to GeneSteps, so I'm going to talk about that later, and the architect is here, Catherine Howell, and they didn't find the gene. Does that mean it's not genetic? No, it is genetic. We just ain't got to the bottom of it yet. So to say that, and I'll come back to this, 50% of our patients we don't have an answer for. So our work in finding the cause is not done, and we have to keep using new genetic technologies and collaborating around the world to get to that. So that's the epileptic encephalopathy. Now we're going to extend the term to developmental and epileptic encephalopathy. And this is where your cause causes this epileptic activity, which causes developmental slowing. But in addition to that, your cause causes developmental slowing in its own right. And we just heard a very clear description of that was I always get the letters wrong, CIYFIP. I should know it, but I'm really bad on that one. And there was a clear history of developmental slowing, and then on top of that you got your epileptic spasm syndrome or infantile epileptic spasm syndrome, and that then is associated with slowing. So that's where you're bringing the two components together. And here's the sodium channel alpha subunit. It could be SCN8A, it could be SCN2A, or it could be SCN1A. And just to emphasise what was said about the other issues, there are so many different features with the DEEs, and this is the gait that we described for Dravet syndrome, and you see it in other of the DEEs, not just Dravet. And there's one of my 27-year-old patients who's got a very severe gait abnormality. We call it a crouch with dystonia. So I'm one of the only two epileptologists in the world I know that see children and adults. And I think that's really important for understanding whole-of-life issues. And this is to emphasise there are many systems involved for the patients with DEEs, many different areas they can have problems. Development is almost always, behaviour is a lot, and you'll hear about that a bit later. Gait problems, gut problems, that can be constipation, it can be not eating, as we heard, sleep and movement disorders. But it can be further. It can involve your kidneys, your eyes, you can get a scoliosis, a bend in your spine. It can involve all sorts of systems. Now, we've heard these are really rare diseases, but are they that rare? And our large epidemiological study looked at this very carefully through Wellington in New Zealand, and we found 1 in 591 children have a DEE. And that's powerful because we want politicians, public health people to fund these, all the resources we need. So 1 in 590 is not that small, so that's important to know. And thank you to Andreas for showing this picture, which is the next stage of my syndrome picture that I started many years ago. And you can see the different syndromes. You can see the mean age of onset of infantile epileptic spasm syndrome, 6 months, and Dravet. You can see if they're generalised or focal, and that relates to their EEG rather than what. And trying to show their most common etiology. Was it structural? Was it genetic, or both? And you can see Lennox-Gastaut syndrome, for example, is both. But as we heard from Sarah, the field has moved so rapidly. In our review in 2016 with Amy McTague and Catherine Howell, we had 60 genes for this. Fast forward to now, and we have more than 900 genes. So it's a very rapidly evolving space. And that means more than 900 different diseases. And so then Amy McTague, who is fantastic at figures, developed these figures. And just to highlight 4 key areas where genes are important: ion channels such as sodium channels, 2A, 1B, SCM1A, etc. ATP1A3, we heard about that, KCNT1. You can see lots of the genes, KCNQ2 here. A second group, how cells signal to each other and metabolize. How do the cells work? A whole lot of important proteins there. Epigenetic regulation, that refers to above the genes, if you will, the genes that control how the genes express. Is this gene turned off or turned on? That's epigenetics, and I don't have time today, but we just published the first study of that in DEEs and showed that we solved 2% of unsolved patients. So that 50% left, we knocked off 2%. You may say that's not much, but if it's your child, it's really important. And then the third part, and we had a nice talk about a synaptopathy yesterday, is the synapse here, where one neuron talks to the next neuron, and there are a whole lot of very important proteins involved in making those two ends of the neurons talk to each other to fire off thoughts, to fire off everything, fire off seizures, and then here you can see SYNGAP1 that we had a meeting about. So what's important, one of the biggest important take-homes here is that every gene has a phenotypic spectrum. And even if you have a gene that always causes something severe, like BRAC1, it still has a spectrum. But in a lot of the genes that we know, we're finding they have a self-limited end, and Andreas has talked about that a fair bit, as well as developmental and epileptic encephalopathies. So overall now we know of 1,000 monogenic genes. What that means is you just need an abnormality of that one gene, monogenic, and you will have epilepsy or you will have a DEE. You might have a mild epilepsy. We know 1,000 genes that just that mutation or abnormality will cause epilepsy. But the DEEs account for more than 900 of them. So we are where most of the genetics has been found, and as I said, 50% are solved, but we're really interested in the unsolved ones, so don't feel unloved. Make sure you're in research so we can keep looking for your cause. So just looking at SCM1A, I'm not going to belabor this because it's been done beautifully by Andreas, but we have GEFS Plus at the mild end, came out of my PhD a long time ago, that syndrome, and one, they are many. So the poster child, the one we know about, is Dravet, and Andreas mentioned that you can have other ones, early infantile ones as well. But so if you look at SCM1A, it's got a host of syndromes, but take a syndrome, take this one, epilepsy of infancy with migrating focal seizures. And we did a large international collaborative study of this in 2019. At the time, 33 different genes led to this syndrome. And half of them were autosomal dominant, a quarter autosomal recessive, and some were X-linked. So just to summarise the point I'm making, Dravet syndrome, more than 90% of patients have SCM1A. Different syndrome, we've solved 70%, but there are 33 different genes. So it's very complicated understanding genes and phenotype. All right, so you want to say, why bother with all of this? The first thing, and I think we heard that beautifully from Mill, is that this ends the diagnostic odyssey. You can move on. It wasn't that glass of wine you had when you were pregnant. Probably was never the glass of wine you had when you were pregnant, but mums worry about these things. And I like to frame the gene finding as the beginning of the answer. It's not the whole answer by any means. It's why you're all here. It also helps us to understand the associated features. The word we use for that is comorbidities. Understand the prognosis. What will my child look like in 5, 20 years? If it's a new gene, there might be 8 people, you won't know. But you'll be able to get an idea. And are they likely to be, is their epilepsy likely to be drug resistant? That means the drugs won't work. How does it help? Well, it helps you to change the treatment today of the drug to the right drug and get rid of the wrong drug. That happens often with Dravet syndrome still. Patients are put on the wrong drug, carbamazepine, Tegretol, and you get them off it and they do better. And also, very importantly for young families, means they can have accurate genetic counselling about their next pregnancies. So finding the cause, that's what the cause is important. It gives you a precision diagnosis. But I think it's really important to marry up epilepsy syndrome and etiology. They're both complementary pieces of information that add to the story. But very excitingly, it gives you precision medicine, or at least the opportunity to head in that direction, and that's what we're hearing about a lot today, and later on from Yuri too. I've put in a lot more about natural history studies because Danielle Williams, sitting right here, asked me to yesterday, and it does follow on a lot of what Andreas says, but it's our natural history study. And just to pick up on what the wonderful Sarah said, Citizen is not a natural history study, just to be really clear. That Citizen is an American system where you pull in the hospital notes, so with a click of a button you get a hospital note. But many of you will have seen when you go and see your doctor, any seizures? No. Stay on the same drugs, see you 6 months. That is not what we are doing. We are assessing the children, we are looking at their morbidities, we are looking at how families are managing so much more. So just to be really clear. But it will take time and effort for you as well as for us. So we now need to describe the natural history of each genetic DEE. So not just Dravet syndrome, but all the other 936 genes. And then we have to look at those different genes and understand the different severities, or different, whether they're gain of function or loss of function. This is critical because we have these fantastic pharmaceutical companies developing new treatments, hopefully game-changing treatments, as we saw in Andreas's videos. But we can only tell if it's really making a difference if you have that natural history data to compare with. We're doing really interesting health economic studies and developing national standards of care. So I was very pleased to hear Andreas said that this study is going to change that, and I guess we need to think about that in a slightly different way to make it do that. And Emma Palmer, who is here already, are you here, Emma? She's coming from Sydney. Oh, there she is at the back, is the most amazing geneticist, and she's talking to you later, and she's really good at leading us in this direction. And I think very importantly, look at psychosocial impacts. So you can say, oh, this is very nice, they like doing what they do, we care about your children, but why does it matter? And here is, to me, the best example of this. This is a precision medicine called an enzyme replacement therapy. So it's not a gene therapy. These children get this precision medicine into their reservoir, into their brain every 2 weeks. And this got published in the New England Journal of Medicine, which I think is the highest rated journal in the medical field. And the red line is what you have to look at. So here you have the natural history of this disease, which is neurodegenerative. So DEEs are bad. This is worse. It kills you within a few years unless you get this treatment. And you can see here that the new drug, this solipinase alpha, reduces this death rate substantially. The children are still progressing, I think, sadly, but that's why we need the natural history study. No regulatory authority, such as the Food and Drug Administration in the US, is going to approve a $3 million drug, for example, unless they have this natural history data. So that's why it's important. And for those who don't have a gene, we still want you in there, because when we find the gene, we'll have your natural history data. So it's forward-looking. All right, now you saw a slide a bit like this that Andreas showed, and I have just done this for Dravet, but I found those slides a bit too confusing, Andreas, because they have the seizures with all the morbidities. So I've split it into 2 pictures. One is just showing the rate of seizure types, different seizure types in Dravet syndrome. I'm not going to belabour it, but around the ages of onset. But then I wanted to do one about morbidities, and I'm sorry, I know most of you don't have children with Dravet syndrome, but it is the one where the most work's been done, which is why we wanted to, and we've both done a lot of work in that. But just to say, here you look at Dravet syndrome, 90% at least have intellectual disability, and here we've shown that over 40% are severe, 20-30% are moderate, and 20% are mild. So that's the future, based on a lot of data. Sleep problems, we studied that with a paediatric registrar, he was a med student at the time, showing that over 70% of patients with Dravet syndrome had sleep problems, but the type of sleep problem differed depending on what age you were. We looked at autism spectrum disorder. Now, that's just part of your Dravet syndrome, part of your DEE, but 40% of children or adults will qualify for that diagnosis. That may help funding. Behavioural problems, common and a huge problem, and you're going to hear quite a bit more about that a bit later, but I often think that after you you never get over the seizures, that's the wrong word. You get used to the seizures. Behaviour emerges as the biggest problem in families' lives. A crouched gait, I showed you that adult, and now there's been a lot of very elegant work on that by many people in the field. And mortality. Mortality is a parent's biggest fear, and our biggest fear too. In Dravet syndrome, it's well established, both here and other groups, 17% of children will not make 20 years of age. More than half of them die from sudden unexpected death in epilepsy, which is basically die in your sleep usually. We think it's seizure-related, we don't know. Some die from brain swelling, which one of my fellows published some years ago after an episode of status. And of course, anybody with intellectual disability has a higher risk of an accidental death. So that's what a natural history study looks like. It looks like this very complex, when did it start, what does it look like? So this brings me to our DEER program, and it's very dear to my heart. And you can see here, Emma Palmer is hiding here behind Deepak and Alex from Sydney. There's our little Sydney contingent, Catherine here, Piero here, Sam over here. So lots of people really wanting to focus on this. And we have set up for our DEER program an Australian Paediatric Neurology Network with a leader in each site. You can't see it because it's gone over, I apologise. And this is what we're really keen to use in terms of the natural history study, and it's taken longer than we all would have liked to get it all right, but we're up and ready to go and advertising. And so these are the child neurologists, and here are the program investigators. And just to highlight, we have health economists, we have scientists, we have our head of our lab, Michael Hildebrand, imaging people, Terry O'Brien, and we're beginning to rope in the adult epileptologists as well. And I'm not going to go through this in detail, but just to say we have people heading the various aspects of this, including our wonderful bioinformatics team at the Walter and Eliza Hall Institute, which is across the road. But just to talk about the natural history study a little bit more, this is really looking at the natural history of a patient with all their morbidities and looking at it at baseline when they come into the study, and then every 6 months to 2 years, ideally yearly. And then we're going to make sure we have review by a neurologist. I think there's some ideas Andreas talked about that could make us a little bit more formulaic, I guess, to get the responses back to people, and I think we should work on that. Neuropsych assessments are critical questionnaires which will exhaust families, and medical records, which is where the citizen comes in. That's fine, we need them, but they're not what we're talking about. And I just wanted to highlight Eliza, who is sitting just there and will be talking to you later. Eliza is a neuropsychologist. She's just finished her PhD in paediatric neuropsychology, and she has two hats. She is the neuropsych coordinator for this program, and she will work with a neuropsychologist we will employ in Sydney and in Brisbane, and we will also look after the West, but we have to figure out the best way to do that. It will depend on numbers recruited. So talk to Eliza. She'd love to see you all tomorrow. She's going to kill me at this because it's a lot of work, but she is very enthusiastic, and you'll hear about the other project in a moment. So what are we going to ask you to do? And thank you, Amy, for all these slides. Permission to get your medical records. Obviously, we're going to get your consent for research. All these questionnaires, DNA, and you can ask Amy, who's just over here, and she's waving at you. Any questions? DNA sample, ideally blood, but we'll settle for saliva if we have to. Neuropsychological assessments with the wonderful Eliza and the interstate people, and then other assessments depending on your child's course, depending on the systems involved. Now, Amy's made me this whole series of slides, which I'm going to burn through The important thing is that QR code, and that will take you to our website, geneticepilepsy.org. And there you will scroll to the bottom and you will click Create New Account, and you will enter your details and click Save. And then it will take you back to the home page. Then you have to go down to the bottom again and proceed to study. This has been 3 years in the making, I have to tell you. It then takes you to the patient portal. You select where your child usually is seen, is that Sydney or Melbourne or Brisbane, which hospital. You register for your child, and if it's your child you have to say their age, but of course we're interested in the adults too. You type in their name and phone number. I should say this is for the world, not just Australia, and definitely for New Zealand. We love our New Zealand colleagues. And then you click Save, and then you get a study information sheet, and each section can be expanded. It's working, Amy, I can't believe this. And then you click to download the PDF of the study information, and then it will say you are paused, because we want to make sure you have the human element. We are not just at the end of a computer. So our team will speak to you to work you through a study and the protocol. And here are just some of our team. We have more in our team, but these are the frontline guys, or girls, sorry girls. And they will be there to ring you and say, hi, my name is Lucy, I work with the team, and this is what we want to make sure you're happy about that. And then once you've spoken, your account will be updated, and you can click back, you can click the person icon, and then you start the hard work. You have to consent, which Amy's told us is way down the bottom of the page. It's like going to a bank or something, isn't it? And then you enter your child and your name, you agree or don't agree to whatever options you want, and then you click the button to sign the form. And for anyone in Europe, this is all through the correct European protocols, because the European ethics are very tight on the privacy.
Sorry.
Okay.
I know who's doing it.
Oh, that's great. Please, thank you. And so your child is now, after all that torture, enrolled in our natural history study, and we thank you. And then you get a patient dashboard. Oh, did I dash through that one too fast? It looks like I did. You enrol yourself as parents. We want you in there as well, because we need you a bit for comparison, and we need you for the psychosocial impacts and all those sorts of issues, the health economics. You add yourself, you add your partner, you select the same hospital as your child, and you complete the form for yourself. We need to put these slides on the website. They're really good. All right. That was before I even took off. No, it was part of it. But basically, I'm going to show you now, I think it's about a 30th, what you're going to hear about in the next 10 minutes. I've got 15 minutes, according to me. In the next 15 minutes, I went to 14 countries, 3 twice and 1 3 times. That was Italy I went to 3 times. And I just wanted to introduce what you're going to hear about later, which was at Great Ormond Street in London, which is where I trained a long time ago. And we were very lucky to get another grant through the Medical Research Future Fund to fund this study. And you're going to hear about it in detail from Genevieve. And Eliza Genevieve is a senior neuropsychologist, works in adults with depression, but very interested in impacts on people and trying to solve these behavioural issues. And the person here is Sophie Bennett, who is first author of the MICE study. And then we also took the wonderful Briley, because she's got a wealth of experience and really informed the process. And you're going to hear from Jen and Eliza later. But just to say where it came from, this MICE study is about a psychological intervention for children of mostly normal intellect or mild intellectual disability, working with the children. And I think Eliza's going to talk about that. Published in The Lancet, and I heard about it about a year earlier, and I knew that the thing that was driving my family's most difficult for you were behaviour often. And there are so few resources for behavioural help, and psychiatric. Waiting to see a psychiatrist is forever. So I really wanted to make an intervention that would work for children with DEEs, which is weekly telehealth, hour with you, or a long-term carer, but ideally the parent. If we're talking adults, it may need to be a long-term carer, and over 4 years of age, and you have to have significant behavioural or the child has to have significant behavioural mental health problems. So you'll hear a lot more, but I just wanted to whet your appetites. So, as I said, you're going to hear very little about really where I went because I didn't have time, but I just thought I'd share a few pearls. This is Professor Renzo Guarini, who is in Florence. Any of you who've put up with one of my ward rounds when I walk in the door with 10 people, I'm nothing. He has 25. So it's like completely overwhelming. And they all wear white coats. I last wore a white coat when I was a medical student, I think. Even when I was an intern, we dropped white coats. So much more formal. And I spent a lot of time with his colleague Simona, who is very interested in children and adults for DEEs. And so we did a lot of research collaboration together. I then changed my mode of transport and went to Rome and went here to Nicola Specchio. Stefan from Paris came for the day, and this is Nicola's mentor, just to show the white coats continued. And this is Marina Trevisano, who is Nicola's protege. And I was what I didn't say was on Renzo's ward round, they had in the ward 4 patients with CDKL5 at once. Now, I don't think any of us would have 4 CDKL5 patients at once. So just to say that very much it was like my ward round on steroids. And so then in Rome, this is at Gesù Bambino, which is in the Vatican City, so where all the action is right now. And this little girl had PCDH19, which is a gene close to my heart. We found that many years ago, and the parents, I asked if I could take a photo, and the parents then wanted a photo with Marina and with me, so that was very special. While I was in Italy, it was actually a different part of the trip. I did this teaching course on drug-resistant epilepsies with 30 young epileptologists from around the world, lots of mentorship, lots of education, thinking about research. This is the town square, tiny little town, 3,000 people. And what did we do? Well, we had our drink, we worked on classification. And you might say classification is very boring, but that's what gets us in the room. And we've just published this paper about an operational definition of DEEs. I've really said it to you, but aiming to underpin therapeutic trials, these precision medicine trials. We need to give pharmaceutical companies the same rubric to be able to use it, and then just emphasising that you must have the epileptic activity, which is affecting development, you must have impact on development, and often we know the gene. And this builds on lots of work from 2017 for the International League Against Epilepsy. That's the world body for epilepsy, and it's the medical body, and it's about classification. And classification is diagnosis, so it affects everybody in the world with epilepsy. But I just wanted to tell you where we're headed. I have a meeting with Nicola about this tomorrow. You've already seen this Dravet picture showing all the different presentations with Dravet syndrome. But just to say, you can have loss of function, which is Dravet and GEFS plus for SCN1A. But SCN1A also has gain of function, which we discovered back in 2017. We have a mum here whose child has gain of function, and that is very different. And why does it matter? Because if you use a sodium channel blocking drug in a child with Dravet syndrome, they will get worse. It's not working, you block it further, it's a disaster. You use a sodium channel blocking drug in a gain of function child, too much action, you dampen it, you can really improve their life. If you can stop their seizures or improve their seizures, you can get better development. So that's why this concept of a spectrum of epilepsy presentations for a gene is critical and is really framing the way we have to take classification further forward. So it might sound boring, but it matters to your children if we get this right. So next little trip, part of the trip, was to Boston with Anne Paduri, and you can see here she's got genetic zebrafish. And when it's one of our genes, it's called DEPTEC5, and when you tap the cage, the zebrafish bounce because they're not normal zebrafish. And so this is another model. You've heard a lot about mice, but there are other models. And I spend a lot of time in Boston visiting gene therapy companies, Praxis and Stoke you've heard about and Encoded. I did go to the Praxis offices. This is Professor Steve Petru, and this is the office. The reason I'm strangling him is there's an office with my colleague Sam Berkovic's name, but there's no office with my name. And the Y chromosome reigns as always. And we've heard a bit yesterday from one of the people from Praxis that they have small molecule platform and antisense oligonucleotide. We're going to hear a lot more about that from Yuri. Different company, same principle. But just to say we're about to start the relutrigine, I don't know how to say that yet, small molecule study in DEEs broadly, and I'm going to talk about DEE studies a bit later on. And we've already had a patient in Australia get the SCN2A antisense oligonucleotide. But I just really wanted to brag about this, which Andreas mentioned as well, Encoded. And this has been led by Catherine Howell, who's just here, and we're together. But you have done the hard yards. And so we were the first people in the world to use this gene therapy. It's a once-ever injection into the brain, as Andreas showed you. And Catherine and her Megan and Kat both helped with it. And I think you did the first one just after I left. And then we were on speed dial wanting to know, and I was with Anne Paduri in her kitchen, there's Anne, and Catherine sent me a second one in, and we were so excited. So just to let you know, this is in Boston, we're hearing about it in Australia. Okay, I've got 8 minutes technically, so that's okay.
Nope.
I've got 8 minutes, I'm timing myself. Is that all right, boss? Okay, so then the last bit...
Do you know what? I'm not going to give you a room with your name. I'm going to give you 5 minutes and we'll add, because you get extra time later. You get extra time later.
Anyway, so the last bit, I went to Boston. Oh, no, I didn't go to Boston. I went to Toronto, and I met up with the Gene Steps team, and we've already heard from Mil how the Gene Steps study was really important, and this is on top of Toronto Kids Hospital, and this is called the IPCHIP, which stands for this long name, but what's important, it's bringing together 4 major paediatric centres from around the world. We have our own Catherine Howell, we have Amy McTague from London, who I mentioned before, Alyssa DeGama with Anne Paduri, but Alyssa is the young leading member from Boston Children's, and Greg Costain, who is a geneticist. So Greg's a geneticist, Alyssa's actually a neonatal newborn specialist, and the rest of us are all child neurologists. So the question this study has done is say, does rapid genome sequencing make a difference? It was published in Lancet Neurology, great journal, and rapid trio genome sequencing in 100 infants. It only looked at babies with seizures under 1 year of age, and it could have been mild epilepsies or severe epilepsies. They showed that this was doable, a turnaround time of 21 days, but if you're in the ICU, they've had a study show you can do it in 72 hours. So I think we have to up our game from 21 days to 72 hours. Most patients were not in ICUs, so the point is this is different. It isn't an ICU study. Most families were very keen to participate, and we had a 43% success rate in this paper, and you'll see we solved about a third of DEEs, and Mil explained that, but a lot of them were self-limited epilepsies, good outcome epilepsies. So did it make a difference? Yes, it did. Immediately changed the game in terms of management, changed treatment in over half, led to further tests, maybe heart or etc. tests, helped us say, your child will grow out of it, your child won't. And also, very importantly, recurrence risk counselling. Are you at risk of having a second child with this? But what we don't know is that rapid diagnosis going to influence long-term outcomes in 20 years' time. I hope it will, I think it will, but we don't know. So this study showed that it's feasible, that you get the cause for quite a lot of patients, 46%, or was it 43%? Sorry. It changes management and will lead to precision therapies. And then yesterday, Catherine ran a study day on gene steps and an information session for parents, and now the 4 centres have got 466 babies, still a yield of 45%. Mostly the child had their own gene, but there were 5 genes that were the top players. And I put this one, PERT2, I left the 2 off, is a self-limited epilepsy, and these can be anything. They can be self-limited or DEEs, and often DEEs, and all have management implications. And just to show that Catherine's project has spawned papers, this is about applying the model to rare paediatric disease, and we've looked, particularly with Emma and her postdoc, to look at the impact on families, and we're doing a lot more work in that space. Just to make Sarah feel better, I'm now on my way home, and I just want to tell you that I had the most amazing sabbatical, but I'm really passionate about growing young clinician-scientists, so that's mentorships, collaboration, research, teaching, friends around the world, and I hope you heard how much Andreas and I have worked together over many years, get new ideas and global impact together. And just so you know, I made 6 pavlovas for all my friends around the world. And finally, thanking you, Geta, and particularly all of you. We couldn't do our research. These are from 2 different Geta conferences. We couldn't do our research without your very intense and committed involvement. We care, you care, we want to make it better. I want to thank you for allowing your children to participate, but also allowing me to show pictures of your children. I think it makes a huge difference, and particularly to acknowledge the inspirational founders of Geta, Sarah and James and her wonderful husband Andrew, Chris and Dave, who are at the back, and Dave's working just here, and of course the inimitable Danny and Danny Williams. Thank you.